Okay, guys, Timmy coming at you again. And we left off where we were talking about metabolism and we got through anaerobic metabolism. So, what I'm going to do for you today is I'm going to explain to you the process of aerobic metabolism. Now, remember, the two are intimately linked, especially when it comes to glucose. So, a quick review. Remember, when a cell needs to take ADP, and then pop on that third phosphate to make ATP so your cells can do what they're going to do. When glucose becomes the most readily available fuel because of insulin, glucose, once in the cell, enzymes of metabolism are going to begin to break those bonds. And when they break those bonds, they release energy. And in this case, it's enough to take ADP that's floating out in the cell, pop on that third phosphate, and recycle it into ATP. And what you have left over is this chemical compound called pyruvate. Glucose 6 carbon, pyruvate 3 carbon. So essentially what you have done is you have broken down glucose in half, and you made some ATP, which is a good thing. And again, I explained to you that when you make pyruvate, pyruvate liberates a free-floating hydrogen ion. So anything that produces free-floating hydrogen ions is an acid. So this is pyruvic acid. Pyruvic acid, once formed in anaerobic metabolism, it cannot diffuse out of the cell and into the blood. So the only place that it's permeable to is the mitochondria. So again, by diffusion, high concentration in the cytoplasm, low concentration in the mitochondria, pyruvate is going to diffuse into the mitochondria. So what we're going to talk about today is aerobic metabolism. And aerobic metabolism occurs in the mitochondria. So this is one mitochondrion. So if you look, and this is important, the mitochondria has an outer membrane, then it has an inner membrane, and then it has a space in the middle, and that is called the matrix of the mitochondria. And between the outer membrane and the inner membrane, you have the intermembrane space. So this is very important. And again, if you recall, I told you that when enzymes act on glucose or fat, they do so by breaking the chemical bonds and releasing electrons. And again, these electrons are high energy electrons. So by breaking those bonds and removing those electrons, you can create energy. Here's the problem. Free-floating electrons are bad for you. They can kill you. So they got to be handled. So you got to remember this. Inside your cells, and especially located in the mitochondria, you have these things, these electron catchers. You have nicotinamide adenine dinucleotide and flavin adenine dinucleotide. Flav. Anyways, so their job is to swoop in when the enzymes break the chemical bonds and to catch those free-floating electrons. And because electrons are negative, they have a negative charge. When NAD catches 2 electrons and FAD catches 2 electrons, the ubiquitously floating around hydrogen ions will bind to those 2 electrons, which are negative, and the hydrogen ions are positive, and you will form NADH2 and FADH2. So the purpose of the Krebs cycle is to break those chemical bonds and catch those electrons. And then along with it, you catch some hydrogen ions. Now look, in the Krebs cycle itself, in the matrix of the mitochondria, in the Krebs cycle, it's nothing more than a series of enzymes that continue to break down what's left of glucose. So there's not a lot of ATP made in the Krebs cycle. What you do make is a bunch of that NADH2 and FADH2. And if they're taken to the right place, they can make ATP. And I'll show you that in a minute. So let's get back. You broke down glucose to pyruvate. Pyruvate can't leave the cell. It goes into the mitochondria. And again, pyruvate's a 3-carbon compound. So when pyruvate diffuses into the matrix of the mitochondria, the site of the Krebs cycle, of course, you can't forget that Neo is in the matrix. There he is. Take the blue pill. Them Duke boys got my money. So we have pyruvate now in the Krebs cycle, in the matrix of the mitochondria. And as soon as pyruvate gets in there, an enzyme is going to act on it and break chemical bonds. Now, if we look, and again, this is important, we look, what's glucose made out of? It's made out of carbon, hydrogen, and oxygen chemically bonded together. So what happens? The enzymes break those chemical bonds, and they release the electrons, and then NADH2 and FADH2 come in and swoop them up. But what you have freed up is you have freed up carbon, oxygen, and hydrogen. And the cells of your body don't like that, these free-floating atoms. So they will combine the carbon and the oxygen to form carbon dioxide, and they will combine to form the hydrogen and oxygen and form water. So these are the byproducts of metabolism, at least 2 of them. So that's how you get the byproducts of metabolism. Carbon dioxide being a waste product you got to get rid of, and water, well, that's made too. So here we go. So we've got pyruvate 3-carbon compound diffused from the cytoplasm into the matrix of the mitochondria. Enzyme acts on it, breaks some bonds in it, and it will form some carbon dioxide. So now you've subtracted a carbon here, so now you have this 2-carbon compound called acetyl. So acetyl is really a critical juncture in metabolism, and I'll explain that in a minute. But now what you want to do is you want to get acetyl ready to enter the Krebs cycle. And again, the Krebs cycle is a series, a circle of enzymes that will break down the chemical bonds that held glucose together, and what's left of glucose is, in this case, acetyl. But here's the kicker, and here's the important thing. In order for acetyl to enter the Krebs cycle, it has to bind with a cofactor floating around in the matrix. I'm sorry, coenzyme. That coenzyme is called coenzyme A. And the main function of coenzyme A is to simply escort acetyl into the Krebs cycle. So acetyl, a 2-carbon compound, is going to bind with coenzyme A, and you're going to have acetyl coenzyme A, or excuse me, acetyl-CoA. So acetyl-CoA is now able to enter the Krebs cycle. And again, the Krebs cycle is nothing more than a series of enzymes that continue to break those chemical bonds and release electrons. So here we go. So now you have acetyl-CoA, and already in the Krebs cycle, because it's a circle, is the 4-carbon compound called oxaloacetate, or oxal. And acetyl is a 2-carbon compound. And acetyl CoA will combine with oxal. And when that happens, the CoA peels off, and then the CoA can be used to escort another acetyl into the Krebs cycle. What you have formed is you formed a 6-carbon compound. Again, the Krebs cycle is nothing more than a series of enzymes that continue to break down these chemical bonds. So now you got the 6-carbon compound, and you got these enzymes in here, right? Enzyme 5. What's their job? To break chemical bonds. And when you break chemical bonds, you're going to release electrons. These electrons are bad, so who's floating around inside the Krebs cycle? NAD and fat. So when you break the chemical bond, release the electrons, NAD comes in and grabs those electrons. Now, you've broken some carbon and oxygen, hydrogen off. So this is where you begin to form more carbon dioxide and more water. Now you got a 5-carbon compound because you broke off a carbon on that 6-carbon compound. And another enzyme, enzyme 6. That comes in, breaks the chemical bonds, releases some electrons, and then who comes in? Fat. Bam. He grabs those electrons. And now you're back to your 4-carbon compound, oxaloacetate. The result is that this little series, the Krebs cycle, can continue. So let's look. If you look at what you made in the Krebs cycle, and again, you don't make a lot of ATP directly in the Krebs cycle. What you make are these electron catchers. So we're looking at the mitochondria, simplified version, right? Outer membrane, inner membrane, intermembrane space. Then you got the matrix, of course, It's like picking a scab, man. You got to do it. So what have you made in the Krebs cycle? Well, you've made some carbon dioxide. You've made water. You've made some heat. And you have caught those electrons. And remember, when you catch those electrons, those hydrogens are going to bind there, floating around, and you form NADH2, and you form FADH2. Now again, you only made a few ATP in glucose to pyruvate. You need more. So these aren't ATP, NADH2 and FADH2. However, if they're taken to the right place, you can make ATP out of them. And that place is a series of proteins that are embedded in the inner membrane of the mitochondria. And it's called the electron transport chain. And what does the electron transport chain transport? You're not going to believe this. Electrons. But here's the kicker. And you need to get this. The final electron acceptor in the electron transport chain has to be oxygen. If oxygen is not present, Krebs cycle and electron transport chain don't work. And that means you don't make ATP aerobically. And then you run into all kinds of problems. So basically what happens is that once you caught these electrons and the hydrogens bound to them, these guys go to the electron transport chain and they drop off their electrons. So I'll draw a little bigger picture of the electron transport chain. So here we go. So you have the inner membrane, then you have the outer membrane. So here is the matrix of the mitochondria, where Krebs cycle occurs. Then you have the intermembrane space. Then you have the intermembrane and the outer membrane. And embedded in the intermembrane are these little proteins. And again, what is the final electron acceptor? Gotta have oxygen. So also embedded in the intermembrane of the mitochondria. There's a series of enzymes. And that series of enzymes, think of it as a little waterfall, like a little pinwheel. You blow on it and the pinwheel moves. So think of this group of enzymes as like a little turbine. So there's like a little wheel in there. And if, let's say, water goes through it, right? So if you have stuff that goes through it, it is going to cause that wheel to spin. So this is how a generator works in Niagara Falls. The water falls down, turns the turbine, and that can generate electricity. So, but you have to have a concentration gradient. You have to have water up high, then it falls down low, and when it goes from high to low, it creates that energy. So again, what did you make in the matrix of the mitochondria? You made NADH2 and FADH2. So these guys are going to dump off their electrons into the electron transport chain. So NADH2 comes in and dumps off 2 electrons. And when those electrons move, when they're shuttled, they're physically moved from one protein complex to the next. So it's like you got a chain of people, a row of people, and you're passing down, I don't know, food, right? You pass it down. So that's what's happening here. Each time those electrons are passed from one protein complex to the next, 2 hydrogen ions that were in the matrix are pumped into the intermembrane space. So you shuttle some more electrons from one protein complex to the next, 2 more hydrogens get pumped from the matrix into the intermembrane space. And then when these 2 electrons, which are negative, bind to that oxygen, 2 more hydrogens are pumped from the matrix into the intermembrane space. Now, when oxygen binds those 2 electrons, floating around in the cell are hydrogen ions. So oxygen has 2 electrons, and it doesn't matter where you get the hydrogen ions from. It doesn't matter. They're floating around. They're ubiquitous, right? And those 2 hydrogen ions will combine with those 2 electrons, and they will form water. So essentially what you've done is you've used up the oxygen. So what have you done so far? Let's recap. You have dumped off your electrons. The electron transport chain has shuttled those electrons from one protein complex of the electron transport chain to the next. But in doing so, you have pumped hydrogen ions into the intermembrane space from the matrix. So now what you have is you have an area of a lot of hydrogen ions and an area of less hydrogen ions. So high concentration of hydrogen ions, low concentration of hydrogen ions. And where do things always go? High to low. Here's the kicker. Once they are pushed from the matrix into the intermembrane space, they can't go back. They're stuck there, at least for now. The only way they can get back is by going through that special enzyme complex, that little turbine inside the embedded in the intermembrane. And that little turbine, that little power station, is called ATP synthesis. So When two hydrogens from the intermembrane space go through ATP synthase, that is enough energy to take ADP that's floating out in the cell so you got adenosine 1Z 2Z diphosphate. That energy of those hydrogens turning that turbine is enough to pop on that third phosphate and produce ATP. And that's exactly what you want. That's the whole purpose of doing this. So when you do that, you are making ATP. What did you need to be the final electron acceptor? You needed oxygen. So that's why it's aerobic metabolism. So this process continues in the mitochondria, in the matrix, and in the electron transport chain. That will continue as long as oxygen is continually replaced, because when those two electrons bind to oxygen, then they're converted to water. So you have to replace that oxygen. That is why if your heart stops or you're not breathing properly, then the amount of oxygen you deliver the cells of the body is going to be less. The result is that you're not going to be able to make ATP aerobically, and that has some profound effects, because if you look, glucose to pyruvate, you make 2 ATP. Not a lot, but you can make it fast. Krebs cycle, more specifically the electron transport chain, you can make 34 ATP. And when you do this, you completely break down glucose. So if you're going to get the most out of the food that you eat, you want to completely break it down. And that means breaking it down aerobically. So what's left over? What are the byproducts of metabolism? Well, you have glucose. And in order to completely break it down, what do you need at the end of the electron transport chain? Oxygen. And what's left over is carbon dioxide, water, heat, and ATP. And that's the whole purpose of it. So carbon dioxide, you got to get rid of. We'll talk about that later. So carbon dioxide is the big waste product that you got to get rid of, and the lungs handle that. So think about it. Think about it. When you are running, you need to contract more muscle. What does muscle need to contract? What does it need? It needs ATP, right? Where do you get the ATP from? Well, hopefully aerobically, right? So when you use the ATP to make your muscles contract, you start building up ADP. So now you got to recycle that ADP into ATP. So if you are using more ATP, you are going to build up more ADP inside the cell. And you're not going to believe this, but the single biggest stimulator of metabolism inside cells is the buildup of ADP. So if the cells are using more ATP than normal, then when they break off that third phosphate, that's going to, when they break that off, that's going to build up more ADP. And ADP tells the enzymes of metabolism to work faster and break down more glucose or fat to pop on that third phosphate. So if your muscles are using more ATP, then they're going to build up more ADP. And what's going to happen to metabolism, or the rate at which you break down glucose or fat inside those muscle cells? It's going to build up. So what does that mean in terms of the amount of oxygen you're going to need? If you're needing to make more ATP, that's going to go up, and the amount of carbon dioxide is going to go up. And you don't know this yet, but I'm just going to tell you. CO2 stimulates breathing. So the more CO2 you produce, the faster and deeper you breathe. That's why you breathe faster and deeper when you exercise. Bam. Okay, now let's get down to the important stuff. Number one. There you go. You got the molecule adenosine, 1Z, 2Z, 3Z phosphate. All right, I'm going to get up and I'm going to run. What do I need to make my muscles contract? ATP. Where do I get the energy? By hacking off that third phosphate, releasing that energy and making my muscles contract, right? So you get that energy. And you get a little heat too. That's why your body gets hot when you exercise. Okay, what do you got that's left over? You got adenosine diphosphate, plus that little phosphate that wants to get hooked up with ADP. So as ADP begins to build up in the cell, and why is ADP building up in the cell? Because you are using ATP. As that builds up, that's going to tell the enzymes of metabolism inside that cell to work faster, to break down more glucose or fat so that I can get enough energy to pop on that third phosphate and recycle ATP so I can continue to contract my muscles. So the biggest stimulator of metabolism is the buildup of ADP. So watch. If you exercise, you will use more ATP. Where do you get the energy to pop on the third phosphate to ADP? Well, you eat. So instead of that ham sandwich being converted to fat and being placed on your hips, butt, and gut, that is used to get broken down inside your muscle cells to pop on that third phosphate. That's how exercise works as weight loss, right? You can lose weight by using more ATP, and your muscles are where you use the most of it, right? Because most of us aren't sitting around doing nothing, I mean, most of the time. All right. So again, what stimulates metabolism inside cells? The buildup of ADP. So here we go. Now, from the clinical side here, how do you get oxygen to the cells of the body? How do you get it there? There are two things that have to happen. Two things that have to happen. Number one, you have to breathe the oxygen in. And number two, your heart has to pump that oxygen down to the cells. So anything that affects breathing your ability to get oxygen into the blood, or your heart pumping is going to affect oxygen delivery down to the cells. So here we'll make a big old mitochondria. Yeah. So let's say, for example, that your heart stops and you're not breathing. If your heart stops and you're not breathing, then you are not going to be able to deliver the much needed oxygen to the end of the electron transport chain. So if your heart stops and you're not breathing, like in a cardiac arrest, for all intents and purposes, aerobic metabolism stops. But you learned that there's a way to make ATP without oxygen, and that is through anaerobic metabolism. So glucose is going to get into the cell. Yeah. Got these little enzymes, and they're going to begin to break the bonds that held glucose. How many carbons is glucose? 6. And you're going to be able to take ADP that's floating out in the cell, pop on that third phosphate, and make ATP. Not going to make a lot of it, but you can make it quickly. What's left over is pyruvate, 3-carbon compound, right? But remember, when pyruvate's formed, what does it produce? It produces a free-floating hydrogen ion. So that's an acid. And that, if it builds up in the cell, is going to drop your pH, destroy your enzymes, and it's over for you. So the question is, can pyruvate diffuse into the mitochondria and get Krebs out? The answer is no way. Why? You ain't got no oxygen here because your heart stopped and you stopped breathing. So glucose to pyruvate is going to continue, but if you don't handle this pyruvate, it's going to be over for you in about 30 seconds. So the body always does stuff that makes sense. So as pyruvic acid builds up, because you cannot send it to the Krebs cycle electron transport chain, pyruvate is converted to a less acidic, membrane permeable acid called lactate. And lactate, when it forms free-floating hydrogen ions. It just forms less of them than pyruvate. But here's the thing. Pyruvate, which is impermeable to the cell membrane, lactate or lactic acid is permeable. So you will be dumping lactic acid into your blood. So what is lactic acid going to do to your pH? It is going to drop it. And this process of glucose to pyruvate to lactate can continue for about 4 to 6 minutes before the pH of your blood drops to 6.8. And once the pH of your blood drops to 6.8, the enzymes of your brain are completely denatured and it is officially over for you. So, what do? Somebody goes down, you check them, right? They don't have a pulse and they're not breathing. So, you do CPR. CPR, again, doesn't restart your heart. What it does is it mimics your heart beating. And you have about 100 oxygen balls in arterial blood. And when the oxygen goes into the cells with normal physiology, the venous side has about 70 oxygen balls left. So there's still oxygen in venous blood. The problem is that the heart's not beating. So what do? You put your hands on their sternum and you compress their sternum. And when you compress their sternum, that squishes the heart. So chest compressions mimic the heart beating. So you have the mitochondria, you have the outer membrane, then you got the inner membrane, of course. And then you have the little proteins of the electron transport chain, right? So now when you're doing CPR and you are pumping around that blood, you will begin to deliver oxygen to the end of the electron transport chain. So what's going to happen? Well, glucose getting broken down to pyruvate and making some ATP, that's going to predominate. That's going to where you make most of your ATP, right? So you make some ATP, right? But now you got some oxygen here sitting at the end of the electron transport chain. So what that means is some of that pyruvate is going to be able to go into the mitochondria, get Krebs out, and electron transport chain out. I'm making this up, just to give you an idea. Let's say 20% of that is now going to be able to get Krebs out. That means only 80% of the pyruvate is going to get converted to lactate. And you will be dumping less lactic acid into the blood. So if you dump less lactic acid into the blood over time, that means it will take more time before your pH hits 6.8. And that means that if you can prevent that from happening, you can get that person to a hospital where they can get shocked or they can be given drugs that can restart their heart and their heart can start pumping some oxygen down to them. And well-performed CPR can keep a person viable for up to an hour. That's why beginning CPR on a person who is pulseless and not breathing is critical. So one more thing about that. Right before this person died, and essentially they're dead, right? What were they making inside the Krebs cycle that they had to get rid of? They're making CO2. So when CO2 gets dumped into the blood, water's in the blood, you don't bleed dust, right? That will combine with water and form carbonic acid. So it is the combination of lactic acid and carbonic acid that lowers that pH. But if you think about it, and I showed you this in class, if you push on somebody's chest, you depress their chest, and then you relieve the pressure, the chest goes up and down. Well, what does your chest do when you're breathing? It goes up and down. So by doing the chest compressions, you also circulate that blood and you can get rid of that CO2. And because CO2 is an acid, you are getting rid of acid at the lungs. That is why, over the last several years, they have kind of gone away from the ventilations, at least in lay rescuer CPR, and have gone towards the compressions, because the compressions mimic you breathing as well. So that answers a couple questions on the metabolism. Remember, I put up part 1. This is part 2. Then I'll have part 3 to answer the rest of the questions. But just so you know, I want all of this, and I want this whole thing explained to me. All right? Good luck and study hard.